We start with this Biotech board. I want to thank the opportunity to moderate this panel with two great researchers. This panel is made up of Federico Ariel, who's present, and the second presentation by Dr. Jurat Fuentes. He recorded his presentation, so the questions will answer, only Dr. Ariel will answer because Dr. Jurat Fuentes won't be able to join us live.
Darlo al doctor Ariel. Federico Ariel.
Dr. Federico Ariel has a PhD in biologic sciences, specialist in epigenetics and non-coding RNA. He's a CONICET researcher of the unit of University of Buenos Aires and CONICET. He has been awarded nationally and internationally. UNESCO 2001 in science, technology, engineering, and mathematics, the highlighted youth, and Bunge and Born Agricultural Award in 2023. He's in the company Biotech by YPF and CONICET between 2021 and 2023. He's a member of the management team of the Argentine Foundation on Nanotechnology and co-presided the Academy of Young Science. In 2022, founded the company Apolo Biotech, devoted to the design of RNA solutions to replace synthetic pesticides in crops. By its innocuity, this RNA technology is more respectful to the environment and people. Federico, welcome.
Y a Gabriela por la presentación.
Thank you, Gabriela, and the organizers. I'm a teacher, so it's hard to sit while I talk. There's some overlap with the next talk. We had planned for the reverse order, but no problem. In Apolo, we do RNA technology. We communicate with plants and provide necessary information so that it can adapt and develop in better conditions. Apolo is a biotech platform. We can modulate plants and pathogens in a non-transgenic manner. We use RNA, and we have our own RNA stabilization technology. Apolo is a spin-off of our science lab. I work at UBA, but it's part of CONICET and the National University of El Litoral. 2021-2022, we received some research grants that were key to develop our technology based on RNA for agriculture. We had successful proof of concept, so we launched Apolo. To date, we had the first partnerships and trials. The developments were recognized worldwide, and now we are an expansion stage with regulations to reach the market as soon as possible. The most important landmark, 2-3 months ago, we started the first pilot plant of RNA production in Latin America. It's in Santa Fe. It supplies the local market for intense crops, and it allows to escalate production and minimize costs. Talking about RNA technology, which is competitive to chemical solutions in the market. It's a reality thanks to this RNA production plant. Our business model is B2B. We have partnerships with companies that distribute our products. It's pre-commercial agreements to develop market-needed solutions, applying scientific knowledge, and reach final stakeholders, the agro producers, through companies we partner with. We have been working on RNA for 15 years. We are devoted to 3 targets: to use RNA to adapt environmental plant adaptation, allow for the accumulation of nutrients and metabolites. Closer to the market, we want to have products registered in Apolo, providing specific immunity to pathogens, replacing pesticides. How does RNA work? RNA is the intermediate molecule between DNA and proteins. RNA, messaging RNA, is to produce vaccines. So we block the infection and we produce immunology. Plants don't have antibodies, so what we do is to introduce a segment of the pathogen, virus, bugs, fungi. It's a double-chain RNA. Plants absorb RNA. You don't need to inject it as a vaccine. And that pieces of RNA acts as a defense barrier. It identifies pathogens and blocks them. It's interference RNA. You block the infection, so it's a vaccination effect, like the analogy in the pharma industry. To take this technology from the lab to the field, we need stabilization. It can be sprayed like any technology in the field, but the RNA molecule is labile. It degrades. So we developed carriers capable of stabilizing RNA to be absorbed by the plant so that it can read that coded info. Based on literature, we knew that this information in the lab was capable of controlling fungi, virus, insects, and currently the only technology based on RNA is to control bugs in the U.S., the Colorado potato bug. And we decided strategically to start with fungi. We can tell you later why, but it is more feasible to reach the market with competitive solutions in the short term. So we were replacing synthetic fungicides with an RNA-coded treatment, and plants protect themselves. Let's show you the proof of concept. This is tomato botrytis. We first work on it because we know it well. Botrytis with stabilized RNA can be controlled in vitro and with inoculation. Control inoculation on leaves and control the infection throughout the plant. This at the lab. Look at the whole plant. Inoculating with 1.5 million spores of botrytis. The plant is perfectly well, no disease symptoms. Interestingly, we were able to prove at the lab that this technology is systemic. On that leaf, we create the RNA, but we protect that leaf and other distal leaves. 3 weeks after RNA application, the plant had new leaves, and those new leaves were protected against the botrytis-controlled infection. Based on this proof of concept on botrytis and tomatoes, we worked on other crops too. the design in the lab and in the field, and now we are working on the challenge of regulating the product with Senasa. So Apolo developed projects in different fungi and crops. Some have great incidents in Argentina, like on peanuts and others. I have other examples here too. Proof of concept in Fusarium. On the leaves it's easy to see. It's not the target organ of the disease, but as you see, it is perfectly well controlled in lab conditions. This is for Sclerotinia minor, and Phytophthora too on tomatoes. You see the prototypes with some specific genes. We fully control the disease under controlled conditions. This is the fungus that creates Sigatoka. We control its growth up to 76% in vitro, and we have controlled trials in the field in Colombia as well. For citrus disease like Penicillium and Geotrichum, it's controlled up to 80 to 100%. We're hoping to bring that to the surface of the fruit to protect it. This is Decaphora that affects peanut smut, and we have field testing that I want to share with you. All that works at the lab. Other pathogens we could not control, but what worked on the lab works in the field. That's what we have proven. There were additional challenges. RNA is a very labile molecule. We are able to prove that at ambient temperature the molecule is saved. It is stable after weeks. We tested it at different temperatures and times. It's being stabilized. That's electrophoresis. If the band disappears, it's stabilized. At a pH range that is broad, it works. But when you come to the field, you have additional challenges. From the lab to the field, an issue. First problem, we come in, the stabilized RNA precipitates. This is precipitating. At the lab, it's not issue because you shake and you spray, but on a tank, that became an issue. So we started working on formulations of the active ingredient so it becomes an emulsion, staying in suspension to be applied in the field. Then, when we got the formulation, the challenge was to see if it was compatible with other chemical agents commonly used in agriculture. Amongst that, of course, are the coadjuvants. The correct water conditions, drop characteristics, but not all coadjuvants are compatible with our technology. We screened it. In which ones we tested RNA is not released, and we went a step further and signed a strategic partnership with Speed Agro to develop specific coadjuvants for RNA technology. In this way, we know that it's compatible and it empowers our active ingredients in the end product. So we are working with them with this compatibility between Ariel and the coadjuvants and other agreements between Apolo and Spidagro. This is with tomato. These are the moments of application. Depending on the phenological status of the plant, we have a chemical control as well, and we were able to obtain results with zero residue technology, the same efficiency with chemicals and without any trace, and it can be applied later on because it's not going to leave residue
grow on the plant or the fruit. This is what happened with strawberry. We had very good results with the flowers. This is Fusarium in tomato. With something new is that we apply the RNA when the plants were still on the tray, so before they were in contact with the soil, with the pathogen. So we are inoculating the plants before they are planted. So that had a very high efficiency. We were assays where the plants that were vaccinated or inoculated didn't have any symptoms. This is the tomato, Sclerotinia minor in tomato. Also the same system in a tray. We had good results. The incidence in that assay was low, so we're going to repeat it because otherwise with a low incidence you cannot control much. And this is Mani peanut in assays together with the Spidagro, where we were able to control very well compared with other. And this is also carbon peanut. We had better results, and with this specific test, this carbon peanut is a very difficult disease to control, but we were able to silence it. But the interesting thing is that we were able to see what was the productivity of the parcels, and the plants treated with RN had a higher level of productivity, and we're thinking about new strategies for technological packages, and such as the quality of the peanut or high quality, and we want to give it this as a vaccine. So that's all I wanted to share with you. Thank you very much. Let's hear the next presentation. I'm going to introduce Dr.
Juan Jurat Fuentes es licenciado en Biología de la Universidad de Valencia.
From the University of Valencia in Spain. After obtaining his degree in the University of Georgia in the United States, he was hired by the University of Tennessee, where he is now professor and director of the Physiological and Pathological Molecular Pathology Department of Plants. The lab directed by him centers his research in developing biotechnology for the control of insects that are more sustainable with the environment for a sustainable vegetable production. His research is centered on insecticides, toxin of Bacillothuringiensis produced in transgenic plants, and silencing of genes for interference genes for plague control. Dr. Jurat Fuentes has published over 100 articles in international magazines, 16 chapters of a book, and he has 2 patents that were granted and 2 pending. More than 100 presentations in international congresses in different places of the world. He has been acknowledged with different awards, international and national awards, as young professor for physiology and insect toxicology in the University of America. He is now the president of the International Association of Invertebrate Pathology. We are going to now listen to his presentation. He cannot be with us. Thank you very much for the presentation and your introduction, for the invitation to participate in this event. Good morning, everyone, and I'm sorry I was not able to attend this event in person. The objectives of my presentation are basically going to be, first, to give you an idea of what is happening with the BT crops and the industry response. I will also share data on experiments in my group to increase the activity and extend the use of BT insecticidal proteins. And then I will focus on gene silencing as one of the next most important strategies for pest control. When I talk about BT, I am referring to transgenic crops of soy, corn, or cotton, commercialized in Argentina but also in other countries. And these BT cultures produce insecticidal proteins, either of vegetative origin, such as the VIP3AA, or that originate from the parasporal crystal, or CRE proteins, of the Bacillus thuringiensis bacteria, or BT. These insecticidal proteins provide effective control of IANA pests when expressed by these transgenic crops. And the use of these transgenic hybrids usually gives rise to economic benefits, since they have a higher production and lower expenditure on pesticides. These transgenic crops, especially in the case of corn, also contribute to the reduction of fungal and mycotoxin infection as a consequence of the damage inflicted by the insects. However, the benefits of this technology from transgenic BT crops are clearly threatened by the evolution of resistance in IANA pests. In the graph on the right, you can see a clear correlation between the years since the implementation of BT crops worldwide and increasing cases of resistance with practical consequences. If we focus only on the Americas, we can see that most cases of resistance affect the United States, most likely because this is the place where these crops have been used the longest. In Argentina, there are currently 3 cases of practical resistance to BT crops that have been reported. 2 of them are from the stem borer, Disertaron sacralis, which has developed resistance to corn by producing the CRE1A toxins and also CRE1F toxin. The third case in Argentina is the late armyworm caterpillar, Podoptera, which has developed resistance to corn that produces the CRE1F protein. However, there are also cases of warnings of imminent resistance to insecticidal proteins in transgenic BT plants. For example, Alejandro Vera at the Obispo Colombres Agroindustrial Experimental Station has already presented the case of Racemplusia soybean meters, increasing the foliation surface of BT soybeans with Quest from one season to the next. As you can see in the figure, on the left is the damage of Racemplusia in 2022, on the right is the damage of the same species in 2023. This increase in foliation means that the frequency of resistance to this transgenic soybean, which produces two insecticidal BT proteins, is growing over time, and practical resistance will likely develop in the future unless alternative control measures are taken. Another example, kindly provided by Alejandro, is the damage and number of spike caterpillars in BT corn producing the protein PIB3AA. As you can see in the graph on the left, while in the 21-22 season no damage was documented by the Helicorpaceae larvae, both in the vegetative and reproductive phases of the corn, the following year the situation changed, with 40% spike with damage and up to more than 2 caterpillars per scythe detected during the reproductive phase of corn. Alejandro and the experimental station have been collecting evidence that will demonstrate that the frequency of resistance to BT insecticidal protein continues to increase over time. So the development of resistance with practical consequences may be imminent. Taking all this into consideration, it is important to develop new control strategies. One of them provided by the industry is the development of new varieties of Bt plants that produce insecticidal proteins active against pests that are resistant to current Bt crops. A clear example is the public-private collaboration between Embrapa and Helix company in Brazil to develop Bt Max corn, which produces the CRI1D protein, which is effective in controlling caterpillar resistant to other proteins, CRI1 and CRI2. Other companies, however, have focusing on transgenic crops that produce other insecticidal proteins that have a different mode of action than the Bt proteins used until now. Many of these proteins have been identified in organisms different from Bt, including bacteria, but also ferns and other plants. These crops are very promising and will surely be commercialized in the not-too-distant future. In our laboratory, we have been collaborating with the company Impetus Ag, researching the possibility of increasing the activity of Cre proteins as a possibility of extending their currently marketed usefulness. To do this, we have focused on the mode of action of these CRI proteins, which recognize receptors in the intestinal cells of the pest insect. Once they bind to these receptors, the toxin forms a pore in the cell membrane, which kills the cells, end up rupturing the intestine, and leads to the infection of the insect's blood and its death. Our strategy has been to design proteins that can serve as a kind of glue or bridge between the toxin and proteins in the insect intestine. In this way, we hope to increase the amount of CRI protein that binds to receptors in the intestine, and thus the formation of more pores for higher toxicity. In our experiments, we have focused on the Cre1F protein as a model, which is produced in corn, soybean, and cotton hybrids, and also in Doptotera frunciperda larvae as the model pest to control. Spotpera frunciperda is an insect that is susceptible to CRI1F. Using this insect, the late military caterpillar, we have been able to verify that the addition of our bridge or glue protein, which we call synergist, considerably increases the toxicity of the CRI1F protein, where those that causes 39% mortality reaches cost the death of 97% of the larvae when we add our synergist. This technology is also useful for controlling the spike caterpillar, Helicoverpsea, which is naturally not very susceptible to the CRI1F protein. With the addition of our synergist, we can increase activity and go from 19% to 88% of dead larvae. In a similar way, our synergist protein can also increase the control of the late armyworm with other toxins, such as the CRI1C, which is naturally not very active against this pest. With the addition of the synergist, we increase the toxicity from 31% to 88%. This technology is still in development, but it can be used to increase toxicity and thus reduce the frequency of insects that survive exposure to Bt protein. Another totally different alternative to those that I have presented to you about other crops against toxins and the synergists is to develop other biopesticides with a totally different mode of action from that of Bt proteins. And as a consequence, it is expected that these new biopesticides will be active against pests that are resistant to Bt. One of these technologies is what I call the gene silencing by RNA interference, or RNAi for its acronym in English. The mode of action of this technology is different from others and focuses on the disruption of the central dogma of biology which is shown on this slide. This dogma explains that the information contained in the DNA sequence, which forms the typical double chain with two strands, is transcribed into an RNA molecule that is then translated into a protein. And the protein is really the molecule that performs the function in the body. Gene silencing by RNAi begins with a double-stranded RNA molecule or dsRNA. This double-stranded RNA will make the translation step impossible, so the protein is not produced, and if the biological function performed by this protein is vital, the process of gene silencing will result in the reduction of the levels of this protein, and thus cause death or an important syndrome in the insect. If we look at this process at a more detailed level, we will find the reason for one of the most interesting characteristics of gene silencing, its high specificity, which results in its high environmental safety profile. We designed the double-stranded RNA molecules so that the sequence is complementary to the RNA target that codes for a vital protein in the insect. Once the double-stranded RNA is ingested by the insect, it travels to the interior of the intestinal cells, where the two strands are separated, and one of them is used as a guide to search for the complementary sequence, in this case our target RNA. Double-stranded RNA is a signal to the cell that the RNA is defective or is a virus, so it must be eliminated. So once the double-stranded RNA strand encounters and binds to the target RNA, the molecule is degraded and the target RNA is not used to produce the vital protein, so the insect dies. Since the specificity of the binding or double-stranded RNA to the target RNA is based on the sequences being complementary, pest control using this engine has one of the highest specificities of all pesticides. In laboratory experiments, it has been shown that when we design a double-stranded RNA for a specific type of pest, it only kills that type of insect and not another one. In the graph on the left, you can see A, B, C, D RNAs designed to kill in A fly, B a raggy, C a beetle, and D a bedbug. As you can see, high mortality is only detected with a specific RNA for that type of insect is used. The specificity of double-stranded RNA is so high that we can design them to kill a single species of fly, even when we use them among taxonomically very close species. That is shown in the figure on the right, where mortality is found only when the double-stranded RNA specific for each species was used. This high specificity is very important because, first of all, it assures us of a zero or minimal environmental or health impact, and at the same time it provides us with a way to design pesticides against pests that are difficult to control. Different methods of distributing double-stranded RNA as a biopesticide have been developed. Obviously, one method is production of RNA in transgenic plants, but there are also methods based on the production of double-stranded RNA solutions that can be distributed systematically through injections in the trunk or in the soil as part of beds, by irrigation and drip irrigation, or even as seed coating. In the United States, there are already two products on the market that are based on this gene silencing technology. One is transgenic corn from Bayer against the corn rootworms, and the other is a biopesticide from the company Greenlight against the potato beetle. In fact, the Greenlight company has made public its interest in continuing the development of this type of biopesticide based on gene silencing against different pest and phytosanitary diseases, from Acarovarroa to botrytis, powdery mildew, fusarium, the fall armyworm, or the beetle pollen, even for the control of those analyzed, such as the butcher's weed, as shown in the photograph below. However, as shown in the right column on the table, most of the technologies are still in the early stages of discovery and laboratory study and are not expected to reach commercialization for a couple of years. One of the limitations of gene silencing for pest control is that it's not effective against all types of insects. For instance, it is not very effective against Lepidopterans, caterpillars, and borers in our lab, we have been able to demonstrate that the injection of a double-stranded RNA gives rise to lethal phenotypes in the late military caterpillar. In most cases, as you can see in the photograph below, we observe in these larvae that have been injected with the RNA a burst of development which results in mortality. However, when we distribute the double-stranded RNA by ingestion, the caterpillars present mechanisms such as degradation of the RNA in the intestine or the sequestration of a double-stranded RNA in vesicles, which prevents the correct functioning of the gene silencing and makes this technology not suitable through ingestion. An alternative that we have been investigating together with the company Agrosphere is the use of mini-cells. These are inert cells, meaning they are not produced. They are produced by the aberrant division of bacterial cells, as shown in the graph above on the right. This division produces mini cells, obtaining in this figure. In the picture below, you can see the mini cells. The basic idea is that the protection by these mini cells and the release of the double-stranded RNA in the interior of the cell allow us to increase the possibility of obtaining a gene silencing that leads to the death of the larvae. In a first step, the research was a follow-up of the double-stranded RNA marked with a fluorescent, comparing when we were with a military caterpillar with a double-stranded RNA in green with protection or naked, compared with the mini cells on the right. In the panel in the left, with the double RNA, we are able to detect the green fluorescent signal minimum in the posterior part of the intestine and not in the insect. Compared when we use the mini cells to distribute the double-stranded RNA, we can detect that the majority of the green fluorescence is detected in the area of the intestinal epithelium of the caterpillar, which means that we are increasing the amount of the double-stranded RNA that reaches the cells, and with such the probability of obtaining toxicity. In fact, results of good tests with larvae of the late army caterpillar using 4 different concentrations of double-stranded RNA, A being the highest concentration and D the lowest, demonstrate a higher level of toxicity against normal caterpillars, shown in orange, but also against caterpillars resistant to the CRONF protein, which are shown in blue columns. This toxicity is not detected when we use empty mini cells, control mini cells, when we use water treatment, or when we use the naked double-stranded RNA. The effect of silencing can also be detected in the survivors of these treatments, which see their development slowed down compared to the size of larvae in the control treatments. As you can see in the photo below, the mini cell treatments, these larvae can barely survive the treatment, but may not be able to complete their development compared to larvae treated with naked double-stranded RNA. Well, this is all I wanted to share with you in this presentation. I hope to have shown you that the protein activity and toxicities of BT can be increased through the use of synergists, and that this technology may be interesting considering the critical period of current resistance evolution to BT toxin. I have also shown you information about gene silencing by RNA interference or RNAi for its English acronym. This is a very promising control strategy with the advantages of high specificity and short residue duration, and that can be combined with other pesticides for integrated pest management. One characteristic of this technology that I haven't told you about is that it works a little slower than other biopesticides. For instance, while BT we can see mortality in a couple of days, for gene silencing we need at least a week from ingestion to see the effects of the biopesticide. However, as I have told you before, we are still at the beginning of the development of this technology, and there are numerous opportunities to improve its biopesticide capacity as well as its way of use. And nothing more before I say goodbye. I wanted to thank the people in my group who have done the work that I have shared with you. Dr. Eva Del Gafar, who has developed the bridge or glue proteins, the synergists, to increase activity of the CRI proteins. Dr. Ibrahim El Shisheni, who is in charge of the project to develop gene silencing to control caterpillars. Of course, we would like to thank Alejandro Vera from the Obispo Colombres Experimental Station in Tucumán for his kindness in sharing some of the data. And I would like to thank Antonella Fiore, Hugo Permiched, and Eduardo Pagano for their invitation and the opportunity to participate with this talk at the Aapresid Congress. I really hope to have the opportunity to attend in person in the future. If you have any question or comment, please do not hesitate to send me an email at jurat.utk.edu, and I will be happy to respond as soon as possible. Thank you very much for your attention, and I say goodbye, wishing you a very fruitful Congress. Thank you very much.
Acá Federico, una de las preguntas dice, ¿bajo qué
What rules and regulations do you use for the foliage product of RNA? The question on regulations is very important. As I said, the only product regulated and registered worldwide is what Juan Luis shown, green light for the Colorado potato virus. It's a biopesticide. That classification, that category at SENASA doesn't exist. RNA doesn't exist in SENASA. It exists in ANMAT because of RNA vaccines. We are doing consultations, we are having conversations with them to see how to classify it. It might go on biochemicals, not as a bio input, but it can help advance with regulation. It depends on relevant regulations because it's a biomolecule and it is produced by enzymatic synthesis. So we're in a constant dialogue with SENASA. Have you tested with any pest insects? We had a first attempt with chicharrita, with the leaf hopper before it became chaos here. It was a strategic decision to work with fungi first, as I said, because it's controlling an insect. We are not going to insect insects and bugs in the field, but with green light you see it. Look at the doses, 10 grams per hectare of RNA. That's very high, 2,000 times higher the RNA that you need for a fungi control. That's why we decided to start with fungi, because we believe we are closer. It's more feasible to control fungi than insect. In the case of the leaf hopper, controlling insects, we were not able to do it. But we have another strategy, control the disease transmitted by the bug and not the bug. You're vaccinated against dengue, so the Aedes mosquito is not a problem anymore. Don't attack the vector. That's what we do in agriculture, but try the plant defending herself from the disease. It's not simple because it's a virus. They are pseudobacteria, but we're working on it. We're interested. That's the closest we've done on leafhopper. You mentioned dengue, but with the dengue issue in the north of the country, do you think it could be useful as a spray applied in area like households and such? That would go hand in hand with monitoring and applications by the municipality. At least in Tucumán, it's done like that. We're getting rid of water containers. If that can turn into a product that can have an impact on public health, it's hard to predict. The development of a mosquito can be controlled with RNA modulation. Putting water on household water containers, well, it's easier just to turn it around and not hold any water in any case. So scientifically, the principle exists. Here they are asking about sprays. No, not a spray. I don't think so, because you need a very high RNA load to feed the insect to block a metabolic genetic pathway. It's not that simple in that regard. We would have to go where the larvae are laid, and the logistics of the application would be too complex. Another question of Lepidopter larvae. How would you apply? Do you do it on the lab, on the diet, on the plant? If you had to design a preliminary study on it, the preliminary study will always be without the plant. Design an experiment to orally administer stabilized RNA. We're thinking of different strategies, not just a spray to control insects. Maybe we can attract the insect to eat something with bait and provide a high dose of RNA. But we're testing with some model systems of bugs, transgenic mosquitoes that become fluorescent. We want something to be a feasible product. The control of the Colorado bug, it's a landmark. No one will forget about the RNA vaccines the same way. But before we get a product in the market, nothing certain yet. More questions are coming in. A serious problem in peanuts is sclerotinia. Have you been able to test it? We did field work. We're analyzing it. We may do more in the next campaign in October. It depends on the incidence. We had RNA, but not always the case. With sclerotinia, we believe that we have something that can work on it, and we are testing on it on peanut. What cases of non-controlled did you see with RNA, and what's the virus variation? What do you mean no control, that it didn't work? Everything that comes out of the lab, it's because showed effect on the lab, and that effect is validated in the field. with coadjuvants and such. With some fungi, we have problems in the lab, Alternaria, for example. But each fungi has 30,000 genes, and you can only silence some. So we're not silencing the corresponding gene. We need to study and analyze, compare genomes. A lot of bioinformatics work from specialists and RNA specialists. We'll keep trying. With Phytophthora, we showed part of the genome to the plant and it didn't work. But using exogenous RNA to modulate the plant helped. It can be long non-coding RNA. It has a function per se, and we use it at the lab. Hopefully one day I'll have a product to show in Aapresid. But for problems caused by climate change, like temperature excursions or droughts, that type of stress, we can adapt the plant by priming it and use that type of RNA. It's not double-stranded to silence, but that type of RNA, we have found and developed some that can modulate the expression of non-transgenic expression with Phytophthora, and it was used in tomatoes. And now we want to try it on some plant species that are phylogenetically associated to tomato, like potato, pepper, eggplant. Another question: how does RNA enter the leaf? That's a big question. In Apolo, we want things to work. We don't know how, but we have questions about how it can be absorbed. There are two pathways. You can use vehicles that the plant absorbs, so the plant absorbs the vehicle and the RNA is released in the cell cytoplasm. The vehicles we use gradually release RNA, and the RNA then is received by the plant. Whether it's done with specific receptors or by diffusion, it's not yet clear. We are trying to figure it out in the science lab because it would give us the tools to design more efficient strategies. What we know is that the evolution theory is that plants are checking constantly what nucleic acids are in the environment to develop defenses before infection. So they absorb RNA and DNA from the environment to protect themselves. That's selective protection. So we are harnessing that capacity of the plants and provide them with the information we want. Another question: have you tested seed treatment? With Dr. Jurat Fuentes showed. Excellent question. Seeds cannot absorb. What Juan showed is the RNA that the bug consumes. But within the seed, it's not easy. There is a hypothesis: when the seed is activated and hydrated, the molecule of the RNA is highly water soluble. We have preliminary tests in that direction, but there are only preliminary lab observations, nothing statistical, nothing. We're interested on crop protection in legumes with Fusarium. It takes long to do the application in the field, So if we could vaccinate the seed, it would be great. And we're working on another project on RNA technology to empower symbiotic relations with rhizobium, bacteria and legumes. It would be nice to apply it on seeds, non-coding RNA from the plant to make the process more efficient.
Al tener tanta especificidad,
Since the technique is so specific, what will happen for foliage products for the management of a complex of diseases or pests? Well, first we need to design the cocktail, and we need to see, okay, what is the chemical we want to solve, that we want to replace. So that is something we understood in the company, to say, okay, you can have an RNA that controls arthritis. So that can be a product in itself. You can have vid, you in vid. There is a period where you cannot apply it near the harvest because it doesn't have any residue. So that can be a product in itself. Now, if we want to replace a chemical that controls arthritis with other fungi diseases, then we need to have RNA for all the diseases, and that depends on each type of crop and system. So our interest is to say not only arthritis, but to see if we want to replace this chemical in this crop, we need to unite the two diseases, and the two applications can be different. Can be a kit where you say at this date you need to apply this part in the crop, and then in this other date this other combination, and then it has to be super precise to see in which stage of the phenological process of the plant you need to apply it to define certain pathogens that are dangerous at that moment. So we need to consider what we want to replace in the market. So to what you just answered, there is another question. The residuality of the product, what is its stability level, and what type of response it induces in the plant, what is the durability in time? Well, those are quite a few questions. It is stable, the double-stranded RNA is quite stable. When we stabilize with our carriers, it's even more stable. We have different assays with different temperatures, even over 50 degrees, which would lead us to guarantee that it's stable for 2 years. And the other questions were stability and durability in time. Well, after the application, it depends on the pathogen. With arthritis, we were able to show that the plant, even with new leaves, in a systemic way, is protected for over a month, but that depends on the virulence of the pathogen and environmental conditions. It's not the same if it rains a lot or not, what is the temperature at that moment. So probably you need to repeat the application, but also considering the crop management, and that we can be competitive in terms of cost, but to also take advantage of the systemic protection feature to diminish the number of applications and become a good competitor in the market. No more questions, just mine. Do you think it can have some kind of effect or non-favorable effect for bees in a service crop where you have a plant that has been treated or infected, has been infected and treated with this product? That is a very good question. The RNA technology, different from peptides and others, has the warranty that is absolutely specific. So that was shown by Juan with the different insects, but what we did was to develop a very good platform with the support of the Sadowski institution, where we can compare in computing studies genoma and specific designs and see that there are no cross reactions. The genoma of arthritis with other fungi, even beneficial fungi, and with a series of crops that are representative of pollinizers, of fishes, birds, other. So we can warranty what we call design security, even if you take the RNA, it doesn't vary because it doesn't fit any part of the genome. And most of the chemicals now in the industry kill the bee, but what Juan has shown us is very pertinent, because by attacking the gene of an insect doesn't do anything to the rest, because there is nothing in the genome to pick up. When we speak, this is for insects. For other type of animals, we have a machinery for the RNA. We carry the RNA all the time. If you eat the tomato with arthritis, infected by arthritis, then we are eating RNA of arthritis. If we have a tomato with our product, then you ingest that product. So the high level of specificity you can see with the inoculation or vaccines. Vaccine for COVID protects against COVID and not hepatitis. So that is very important when you have emergencies, because the level of efficacy of the product has to do with the complementary section of the RNA. That is what's happening with the peptides, and that is something that will happen with the RNA, because it's similar to the vaccines against the COVID. If you remember, during the pandemic, you had the Omicron variant, and the first vaccine was the Moderna, because it was sequenced, and then I was able to sequence and to change my vaccine. The same we can do. If at a certain moment there is a fungi that is resistant to the RNA treatment, we can isolate it, we can see where the mutation took place, and to redesign it immediately without thinking, compared to a chemical, of new variants, of new changes, molecular changes. Just simply the same production pipeline, the same infrastructure. The only thing we change is the sequence to adapt ourselves and have an evolution with the pathogen. No further questions. Thank you very much, and see you next time.